Hey, hi everyone. Sorry I'm not there today. I have to do some training, but I'll still just go over our last lesson for this unit. Uh, so this is about alternative sources of energy. So instead of getting all of our energy in the form of ATP from carbohydrates or in the simplest form of glucose, we also consume fats and proteins, and we can harness energy from those as well. Okay, we can get ATP from those macromolecules as well. We also can do get ATP from nucleic acids, but that's only in dire situations when we desperately need ATP. Okay, so while carbohydrates are the easiest and quickest macromolecule and kind of like the most standard, for cell to use in aerobic respiration, our body also utilizes other macromolecules for cellular respiration when glucose and other carbohydrates are unavailable. Okay, so remember, our body can store glucose in the form of glycogen in those long chains. You have to copy this down, but remember, we have the long chains of, sorry, that was an O, of glucose with the alpha bond there, the 1,4 glycosidic bond, and then sometimes we'll have branches up to here. That would be to carbon 1. Okay, so that's kind of the base form of glycogen and can keep going on in both directions. Okay, so our body will break down our glycogen stores when we need energy initially, which is mostly found around your liver. And that's why if you have too much, sometimes you can get a little bit of a belly like I have. So our body will break down these reserves first because it can process glucose easiest. It's glucose readily available for us. But when we deplete those stores, that's when we begin to consume other macromolecules. The first one primarily being fats. Okay, so triglycerides are broken down into the glycerol and the fatty acids. So remember, triglyceride has our glycerol, 3-carbon backbone with the ester bond. Oh no, that's C. Oh, sorry. O, the C, double bond O, and then we have the fatty acid chain. Right? Uh, Let me erase this. Okay, so those are our triglycerides. So we break it down into the glycerol molecule and then the fatty acid. Okay, and then each of these does something different. So glycerol undergoes the process called glucogenesis and gets converted back into either glucose if we have two glycerol molecules or G3P if there's only one. Okay, so remember, glycerol is a 3-carbon molecule. That's why it's easy to make it into G3P, which also is a 3-carbon molecule, or into glucose, which is a 6-carbon molecule. We just need 2 of them. Okay, and then it'll enter respiration from glycolysis, either at the beginning of the glycolysis at step 1 if we have glucose, or at coming in at step 5 with G3P. Okay, so we kind of skip the energy investment phase. So then, that's what happens to the glycerol. The fatty acids undergo a process called beta oxidation in the matrix of the mitochondria. Okay, so this process strips away 2 carbon acetyl groups from the carboxyl end of the fatty acid, and then it adds the CoA enzyme to form acetyl-CoA. Okay, and then that enters the Krebs cycle. So what we'll have is on the carboxyl end of the fatty acid, we have coenzyme A attached to it. And then what happens is it kind of happens in 2-carbon intervals, okay? So these 2 carbons, carbon 1 and 2, will be broken off and then used as acetyl. And then it starts up to the next 2 carbons, 3 and 4 here. Split it there. This will come another acetyl-CoA, and then it moves on to the next one. So like in this example, it made 3 acetyl-CoA groups. We just add the CoA enzyme to the end of each of these, the fatty acid. Every time it gets shorter, we put on another CoA enzyme just to keep the process moving. Okay, and that's how it breaks it down our fatty acid. It breaks it down 2 carbons at a time. So that's why our remember back in unit 1 when we were talking about lipids, fatty acids always have an even number of carbons. It's for this purpose. Okay, because when it gets broken down 2 carbon intervals. Okay, and we make acetyl CoA from that. Okay, acetyl CoA is the end product of pyruvate oxidation, and it's one of the first components of the Krebs cycle, along with oxaloacetate, which was the end of the carbon cycle, or sorry, the Krebs cycle. Okay, so we make acetyl CoA, and then acetyl CoA starts into respiration at the Krebs cycle. Okay, now what happens is we get a lot of acetyl CoA from each of these fatty acids, and there's 3 fatty acids. Okay, we don't have to worry about this so much. It's just specifically how it's breaking down a double bond up in unsaturated uh, fatty acid. So just that's the process. Okay, breaks it down in 2 carbon intervals to make acetyl CoA each time. Okay, so when fats are completely metabolized, they yield twice or more than twice the amount of energy as a carbohydrate. Okay, because when fats are cleaved, this cleavage here, uh, here, uh, when they're cleaved, they reduce NAD plus and FAD molecules. Okay, and then those go into the electron transport chain and chemiosmosis to generate ATP. Okay, so they'll reduce NAD to NADH and FAD to FADH2. Those are our electron carriers. They go into the electron transport chain and create ATP through chemiosmosis. Okay, and then the acetyl CoA also goes through the Krebs cycle. Okay, so we're kind of making ATP both from the electron carriers and from the acetyl CoA. Okay, so the larger the fat, the more energy that can be harvested because we're going to make more 2 carbon compounds. Okay, so if we have like a 12 carbon fatty acid, that's going to make less energy than if we had a 24 carbon fatty acid because we have twice the amount of carbons and twice the amount of acetyl CoA that we can produce. Okay, so that's why you may have seen this comparison before, either in elementary school or if you took the grade 12 health science, the 4M. So 1 gram of carbohydrates yields about 4 calories. 1 gram of proteins yields about 4 calories. 1 gram of fat yields about 9 calories. Okay, so more than double the amount of calories, which calories are just a way for us to measure potential energy without using that kilojoules per mole unit that we've been using before. Okay, it's just the general way for the general public, an easy way to kind of digest, if you pardon the pun, how much energy we're going to get out of this. Okay, so calories aren't bad. Okay, calories are just potential energy. So speaking of proteins, when we consume proteins, we can also get energy and from proteins, and they'll enter the cellular respiration stages at different stages. Okay, so proteins are first digested into amino acids, where the amino group at the end. So remember, an amino acid, we have just generally an amino group with our central carbon with some kind of R group that dictates what amino acid it actually is. We'll have the hydrogen up here, and then we will have our carboxylic acid on this side. Okay, so they get broken down into amino acids, and then this amino group gets removed through the process of deamination. Okay, so we're kind of deaminoing the amino acid. Okay, so when it gets deanimated, it picks up a hydrogen and becomes ammonia. Oh, sorry, just let me. I was going to remove that moment. I'll say the call. Thank you. I did remove it. Okay. So sorry, if you want to go back and just pause it so that you have that structure, you can. So we get ammonia, which will kind of go down to our kidneys and get filtered out through there and get converted into urea, which we urinate. Okay, so that's how ammonia gets out of our system. Uh, so it's a waste product for animals and plants, but it can also be beneficial for plants because plants do need nitrogen. Okay, so if plants really send to the soil, they can harvest it back because they need nitrogen, as we'll see in photosynthesis. Okay. Oh, so I forgot I had this diagram here. So we use hydrolysis. We also reduce uh, an FAD. So the NAD gets taken off, and we get ammonia with. Uh, oh, sorry, I forgot to put this in. Oh, it's here. Okay. But one of the things we can potentially get is alpha ketoglutarate. Okay, which is our 5 carbon. One, two, three. Oh, wait. Oh, sorry, that's alpha keto acid. Okay, but this can become alpha ketoglutarate. Okay. Uh, okay, so depending on what type of amino acid we have, remember we have 20 different amino acids. It will enter cellular respiration at different parts of the pathway, okay, which you'll see on the back of your note, okay? So leucine, for example, leucine is a smaller one right here. So this leucine will become acetyl-CoA. Okay, so we remove our NH, becomes ammonia, and then this will become acetyl-CoA. Okay, alanine here we have 1, 2, 3 carbons. It'll pick up 2 more, and it'll become the first 5 carbon compound in Krebs cycle, which is, if you don't remember, alpha ketoglutarate. Okay. Oh, sorry. Alanine turns into pyruvate. 3 carbons. 1, 2, 3. It's proline. Proline. Oh, there's something wrong with that. Yep, here we go. Proline. We have an ammonia in here, kind of in a ring, but then the ring, we get 1, 2, 3, 4, 5 carbons, and that becomes alpha ketoglutarate. Okay. So we can have pyruvate, we can have acetyl-CoA, or we can have alpha ketoglutarate. Okay. So, and they're all going to enter cellular respiration at different stages. Okay. So acetyl-CoA will go into the Krebs cycle. Uh, Proline will also go into the Krebs cycle, but kind of halfway through the Krebs cycle. And then pyruvate will enter in right at pyruvate oxidation. Okay. So most commonly, what we consume and we kind of recognize as receiving energy from are uh, carbohydrates, fats, lipids, and proteins. Okay, that's where we get all the calories from. We can also use nucleic acids in a respiration in times of great necessity. Okay, even though the food that we do does have DNA and it has nucleic acids in it, we don't typically break that down. Okay, because our even though we can consume the food and we can have those nucleic acids and then we don't need them, we have our own genetic information already. Uh, it's kind of a bad oh my god, what's the term I'm thinking of? It's like law, past practice. It's a bad it's a bad thing to start. Okay, because if your body can start using nucleic acids as fuel, then it might try to use it more often, and then you're going to creep into your nucleus and you're going to break down your own DNA, which you don't want to have happen, or RNA that you desperately need as well in your body. Okay, so we just in as a rule, our body goes, no, nucleic acids are sacred. I do not want to break that down. The only time we would ever digest nucleic acids are during times of extreme starvation. Okay, so if you deplete all your carbohydrate, resources. You deplete all of your fat resources. Okay. Or if you consume like most of your proteins and amino acids and you have enough left just to function, then your body might go, okay, we got to start breaking down some nucleic acids for energy. Okay, so this is the last resort. So nucleic acids are broken down into the nucleotides, the sugar, the phosphate, and then nitrogenous base. And then the nucleotides get funneled into the Krebs cycle. Okay, and we'll also produce NH3. Okay, so we'll make more ammonia and urea. But it's bad because this could lead to kidney damage just because the nitrogen bases are very nitrogen rich. So we're going to make a lot more ammonia than we would off of proteins. And then that ammonia could create ammonium in your kidneys if it's in too high of a concentration. Going to lead to kidney damage. Okay, so this is the diagram that we have on the back of your note. It's just showing where everything can enter the cellular respiration. Okay, so this one is a standard. So we go from simple sugars, go through glycolysis to pyruvate, acetyl-CoA comes into the Krebs cycle, and we are going to make water as a final product. Okay, so we have glycolysis, pyruvate, through pyruvate oxidation, we get acetyl-CoA. So we're starting with glucose. Starting with glucose. Glycolysis turns it into pyruvate. Pyruvate goes through pyruvate oxidation to create acetyl-CoA. That enters into the Krebs cycle. And then through the electron transport chain, we are producing H2O. Okay, so polysaccharides are just a bunch of simple sugars, a bunch of these glucoses all bonded together. We break them down to our simple sugars, and then that is the cellular respiration that we've been talking about for most of the unit. The alternates that we have here, I'm not trying to do this in blue because it's still okay. Our lipids get broken up into glycerol and fatty acid. Glycerol undergoes glucogenesis, which can become glucose or pyruvate, but it'll enter into glycolysis. The fatty acids undergo beta oxidation, and they will become acetyl-CoA. Okay. Also from Krebs cycle, we also make CO2. Okay. Our proteins undergo deamination, and then they could become pyruvate, they could become acetyl-CoA, they could become something else in the Krebs cycle. Okay, but then they are going to make NH3. Nucleotides, don't know why this is right over here. So proteins get broken down to amino acids, undergo deamination. Nucleic acids get broken down to our nucleotides, and then this is the bad one that we don't want. They will enter in at the Krebs cycle, but we're already skipping a lot of the process, which isn't good. And we're also breaking down our genetic information, which we desperately do not want to have happen. Okay, and then we're making high concentrations of ammonia. Okay, so that's how we can have our other biological macromolecules kind of enter into cellular respiration. Okay, so glucose is our standard. It's kind of how it normally wants to work. Our lipids will jump in at glucose or pyruvate, and as well as acetyl-CoA to go into that Krebs cycle. Proteins can enter in a bunch of different stages depending on which amino acids we have, and then only if like your body recognizes, hey, I might be dying, I need energy, that's when we're going to use nucleotides. Okay, so that is the end of this unit. I know some people are probably very happy that we're done with cellular respiration. So that's it for today. You can use the rest of the day to start studying for the test. I have the review on U3-L8. But if you have any questions, you can reach me by email, and we'll be having the test on Tuesday. No, sorry, Wednesday. Tuesday's for the grade 9s. Wednesday. Sorry, don't be scared. Wednesday is your test day. Okay, and I'll have answer any questions you have when I'm back on Monday. Have a great weekend.